Hi guys, so today we're talking about cell cycle controls and the types of mechanisms that actually are controlling the cell cycle from happening. The frequency of cell division depends on how old the cell is and what type of cell it is. Something like a skin cell is going to divide much faster. It divides approximately every 2 weeks, versus something like the external muscle around the intestines divides every roughly 16 years. These differences result in the regulation of the cell cycle at the molecular level. Then we have something like cancer cells that have the ability to escape all cell cycle controls and divide uncontrollably, which is actually the textbook definition of cancer is just uncontrolled cell growth. There is evidence that some of these signals come from the cytoplasm. And the cell cycle appears to be driven by a specific chemical signal that is present within the cytoplasm that tells the cell when it's time to move to the different stages of the cell cycle. Evidence for this hypothesis comes from an experiment when cultured mammal cells at the different phases of the cell cycle will merge together to see what would happen. So when we took a cell in S phase or synthesis, it's replicating its DNA, and a cell in G1, and we merge them together, we found that that cell in G1 jumped right into synthesis. But when we took a cell that had already replicated its DNA and was beginning mitosis, and a cell in G1, we found that that G1 cell jumped right into mitosis despite the fact that its DNA had not been replicated yet. There is a cell cycle control systems. It's a sequential series of events that occurs within the cell, and it acts as a biological clock for the cell. It's regulated by both internal and external signals with specific checkpoints where the cell must stop in the cell cycle until a go-ahead signal is received. So our main checkpoint is this G1 checkpoint. If the cell does not pass this checkpoint, it enters a different phase, which we'll talk about in a moment. But the most important one is right here at G2. That checkpoint makes sure that the cell is twice its original size, its DNA is properly replicated, and that it is ready to enter mitosis. And then we have an M checkpoint right here that makes sure that mitosis is fully complete before cytokinesis can divide the cytoplasm. For many cells, though, the G1 checkpoint is the most important because it controls the rest of the cell cycle. If a cell receives the go-ahead signal at that G1 checkpoint, it moves through synthesis, G2 and M, relatively quickly and divides into two new cells. However, if that cell is not ready to move through and it does not receive the go-ahead signal, it exits the cell cycle and enters something called the stage called G naught. Cells can go from G1 into G naught and then back into G1 when they are ready to divide. They won't go back and forth 100 times. They'll sit in G naught or G0 until they are ready to divide. G0 is just a working phase. It's non-dividing, so they stay in that until they get the go-ahead signals. And we can envision this as like a traffic light. If they hit the go-ahead signal, they get a green light, the gate goes down. If they don't get that go-ahead signal, it's a red light, and they sit in G naught until they are ready to get that go-ahead. The cell cycle clock is controlled by cyclins and cyclin-dependent kinases. These are two types of regulatory proteins that are involved in the cell cycle. Cyclin-dependent kinases, their activity fluctuates throughout the cell cycle because of its control by the cyclins. And they're named because of their concentrations, and they vary within the cell cycle. We have one very specific cyclin-dependent kinase, and it's called a maturation promoting factor, or an MPF. This is a complex that triggers the cell's passage from the G2 stage into the M phase. And we can see that MPF activity here in orange, it spikes during mitosis and then dips back, spikes, dips back, versus our cyclin concentration starts to increase at the end of synthesis right before G2, and and it drops off relatively quickly in the middle of mitosis to repeat that process over again. And what ends up happening is that we have our cyclin-dependent kinase at the start of G1. It moves through the cell cycle until G2. When a cyclin comes in, it makes that MPF that then gets broken down during mitosis, leaving the cyclin-dependent kinase in the cycle. These are stop and go signals that have both internal and external signals at their checkpoints. An example of an internal signal is a connectochore that does not properly attach to a spindle fiber that delays anaphase from happening. If the connectochores are not attached to the microtubules properly, we will not move past metaphase into anaphase. Some external signals are growth factors, and these are proteins that are released by certain cells that stimulate the division of other cells around them. An example of this is a platelet-derived growth factor. This stimulates the division of fibroblast cells in human cells, specifically in culture. And what happens is we took that sample of connective tissue and cut it up real small. It was digested, so just the cell was left. Without that growth factor, the cell did not grow, but with the growth factor, it filled up the rest of that vial. This is a clear example of an external signal that is called a density-dependent inhibition signal. This stops overcrowded cells from continuing to divide. Most animal cells have the ability to exhibit anchorage dependence, in which those cells must be attached to a substratium in order to divide. Cancer cells have the ability to surpass both of these external signals, exhibiting neither density dependent nor anchorage dependence. So this is a picture of like anchorage dependence, and what happens is as long as these cells are anchored properly, they will begin to divide. Here's density dependent. What we did is we removed with a scalpel a few cells and noticed that those cells continued to divide. This experiment was completed in the same exact steps with cancer cell, except they realized that those cells continued to divide and stack on top of each other. This creates something that we know is a tumor. Cancer cells do not respond normally to the body's control mechanisms for the cell. Cancer cells don't need growth factors in order to grow and divide. They may even have their own growth factor that bypasses that of the normal cell cycle. They may convey a growth factor signals without the presence of the growth factor, or they may have abnormal cell cycle control systems, and that switch is basically off, allowing them to constantly divide. When a normal cell gets converted into a cancerous cell, it's called transformation. Cancer cells are not eliminated by the immune system and then form tumors. And these are masses of abnormal cells within otherwise normal or healthy tissue. Abnormal cells remain at the original site and may not be cancerous, and we call this a benign tumor. But malignant tumors are those that invade the surrounding tissues and metastasize or move cancer cells from one part of the body to the other. This can form the additional tumors, and the number one type of metastasized cancer that we think of is metastatic breast cancer. It's the movement of cancer from one part of the body into the breast. Recent advantages to understanding the cell cycle and cell cycle signaling have led to advanced cancer treatments. We have radiation, which isolates the cancer treatment into the area of just those actively dividing cells. But we also have chemotherapy. This is a universal cancer treatment, which travels throughout the whole body. And the only real downside to chemotherapy, although it works very well, is it kills all actively dividing cells. And that's why immune system is weakened, hair and nails. Nails are weakened and then hair falls out, because those are all very rapidly dividing cells that chemo is stopping from dividing in order to kill that cancer cell.